Mitosis and cytokinesis. Mitotic phase. Mitosis involves a complex series of stages where the single nuclear membrane is broken down. The DNA forms into tight chromosomes and are evenly separated into two, and a new nuclear membrane forms around each. The division cycle is complete when cytokinesis occurs and two separate cells are formed and the cells enter interphase again. Mitosis. The splitting of the nucleus in mitosis occurs in four basic steps: prophase, metaphase, anaphase, telophase. Because of all of the processes that occur at the end of prophase, a fifth step termed prometaphase is often used. The AP Biology standards do not require memorization of the step names, but you must know the processes, so we will use these 5 stages when discussing mitosis for clarity. The first step in the mitotic phase is prophase. In prophase, the nucleolus disappears, chromatin condenses into chromosomes, which appear as two sister chromatids joined at their centromeres, which you can see here, these centromeres. Centrosomes start to travel to the opposite ends or poles of the cell. So you can see a centrosome here, and they're traveling apart. Arrays of microtubules called spindles start to form from two centrosomes. You can see the blue spindles starting to form. The only time that DNA is visible in a microscope as distinct chromosomes is during mitosis. Tightly packed chromosomes are essential for the equal separation and sorting of DNA into two identical nuclei. So you can see those X-shaped distinct chromosomes. Prometaphase, or the end of prophase. At that point, the nuclear membrane disappears, so you can't see it anymore. The spindle fibers are nearly completed and ready to provide a scaffold for chromosomes to travel. So you see those spindles. Each chromosome attaches to a separate spindle at their kinetochores, a protein structure at the centromere region of the sister chromatids. So you see how these chromosomes are starting to connect to the spindles at their kinetochores. Centrosomes versus kinetochores. This is an image of a human cell during division. Centrosomes in green form a point of attachment at the poles of a cell, and the spindles form out of the centrosomes. The chromosomes appear in blue, and the kinetochores at the center of the chromosomes are in pink. Here's a closer look at centromeres and kinetochores. So we have a chromosome here with two sister chromatids. This is the spindle fiber that attaches at the kinetochore. This entire thing is the centromere region. Metaphase. Metaphase happens after prophase and what we've called prometaphase. At this point, the spindles are completely formed, as you can see, and the chromosomes align on what's known as the metaphase plate, or the equator of the cell. Following metaphase is anaphase. During anaphase, sister chromatids separate from each other. See how they're separated here? They separate at the centromere, and spindle fibers are used to move each sister chromatid toward the opposite pole. So these spindle fibers pull them towards this pole on either side of the cell. In actuality, the spindle fiber is not pulling the chromatid. Rather, the kinetochore, where the spindle fiber connected, is eating or winding up the spindle fiber to move the chromatid toward the pole. Telophase. During telophase, cell elongation continues, because you can see it's becoming more and more oval in shape here as it elongates and stretches out. The nuclear envelope reappears around the chromosomes, which you can see got these blue dashed lines. Nucleoli reappear, so there's a nucleolus in each. And the chromosomes begin to unwind into chromatin. Mitosis is complete at the conclusion of telophase when a new nuclear membrane forms around each set of chromosomes. But cell division must continue to form two separate cells. Cytokinesis. Following telophase, the cytoplasm divides in a process called cytokinesis. Cytokinesis means cell movement. Each daughter cell must move its own components into its own separate space. Cell division is not complete until each daughter cell has formed a complete cell membrane, totally enclosing the new cell, isolating it from its sister cell and the environment. In an animal cell, we get this cleavage furrow here that gives this like its 8 shape, and this contractile ring that forms to eventually separate the two cells. In a plant cell, it's a little bit different. We get these Golgi vesicles that form, that eventually make a cell plate that then allows two separate cells. Because plant cells must build a cell wall in addition to completing its cell membrane, the process of cytokinesis is very different between plant and animal cells. Let's take a look at cytokinesis in animal cells. A ring of microfilaments forms a contractile ring out around the outside of the cell. You can see this contractile ring right here. The ring forms a cleavage furrow here, which splits the cytoplasm in two. The contractile ring uses actin filaments and a type of myosin, similar to what is found in muscle contraction, to pinch off the two cells in the middle. Now let's look at cytokinesis in plant cells. During the G2 process of interphase, the Golgi apparatus store up cell wall material such as glucose in vesicles. During cytokinesis, these vesicles collect in the center of the cell, here, and then fuse together, forming a cell plate. The cell plate forms along the equator of the cell, growing from the inside out and eventually fusing with the outer cell wall here and here. As the cell plate forms, the fusing of the vesicles also build the new membrane. As the new cell membrane forms on each side, enzymes use the glucose brought in by the vesicles to construct the new cell wall in between them. Once the membrane on each side is assembled with cell walls in between, the process is complete. Here's a plant and animal cell comparison of cytokinesis. The purpose is to separate new cells into distinct compartments in both. The major structure formed in the plant cell is the cell plate. In the animal cell is the cleavage furrow. The cell membrane in a plant cell is built along the cell plate, and in the animal cell, existing membrane constricts, pinching off or separating the cells. The cell wall is only formed in the plant cell, and the new cell wall is formed between new membranes. The result for both is two new identical daughter cells.